After running an alignment, the project will open in the NextGENe Viewer. From there, many different reports can be viewed and exported. This is the NextGENe Viewer. It displays a lot of information all at once. Mismatches are highlighted. Novel SNPs are in blue, known SNPs are in purple, and mismatches not passing the mutation filter are gray. The coverage in gray and annotation is shown at the top in the global view. Blue lines are genes, green are mRNA, and gold are coding sequences. Zooming in and out and moving around is as easy as clicking and dragging. The blue plus sign shows the current position in the global view. Reference and consensus sequences are displayed along with the annotation in the local view. Reads are shown aligning just underneath the consensus amino acid sequence. The mutation report can be seen at the bottom here, although it can also be hidden or displayed on the side. A bed file is needed to tailor the mutation and expression reports to the regions of interest. It is a tab-limited text file with 4 entries for each region: chromosome name, start position, end position, and region name. For best results, the bed file should not contain any overlapping regions. If 2 regions overlap, they should be merged and counted as 1 region. A bed file, or a list of capture regions, is usually supplied as part of the sequence capture process. The bed file must be properly formatted to work with NextGENe, and the position of the regions must be for the same reference build used for alignment. The expression report is useful for finding the coverage in any region. To open the expression report, simply select the expression report from the Reports menu. Load a bed file, and click OK. The expression report can be saved as a tab-limited text file by clicking on the Save icon. It can then be opened in a spreadsheet program such as Microsoft Excel for further processing. The expression report lists a segment description, start and end sites, length, and several different measures of coverage. Max counts is the highest single base coverage within the region. Average counts is the average depth of coverage for the region. Less than 1 means it is only partially covered. Read counts is the number of reads in that region. Forward read counts is the number of reads lined in the forward direction, which gives an idea of read balance. RPKM is reads per kilobase of exon model per million mapped reads, which is a normalized measurement. This information can be used to calculate important measurements for capture data, like the number of regions with very low or zero coverage, the average coverage, and the specificity. The specificity of capture is an important measurement to give an idea of how successful the capture was. Generally, it can vary between 20 and 90%. It can be quickly calculated by adding the number of reads, listed as read counts, in each region of interest, and dividing that by the total number of aligned reads. The mutation report setting should be adjusted before exporting the mutation report for the region of interest by clicking on the button shown here. Many options are available, including reporting variations in different nomenclatures, applying several different filters, and displaying or hiding various information. Hiding or showing columns in the mutation report, including allele percentage, mutation score, and coding sequence. The available filters are shown here. Synonymous, reported, low scoring, and non-coding sequence mutations can be hidden. NextGENe produces a mutation score for every variant that passes the mutation filter. The score is based on coverage, variant percentage, and 4 other factors. These 4 parts of the mutation score can be disabled, and often should be disabled because of the data. For example, if most reads aligned in one direction, the read balance score should be disabled because it would only lower every mutation score uniformly. If a score is disabled, on the Scoring tab, it is set equal to 1 for every mutation. If the scoring filter is used for that score, no reads will be filtered out. To export the mutations found in the regions of interest, select Output the Points of Interest from the Mutation Report menu. Filtering by the mutation report settings will give the report the same settings as the full mutation report. As mentioned earlier, there are a few optional steps that can improve data analysis: running an initial alignment and using the condensation tool. Running an initial alignment of the data can be helpful as a quality control step. There are several properties that will vary between capture systems and from sample to sample. The first of these properties is forward-reverse read balance. In whole genome data, you will ideally see half of the reads aligned in the forward direction and half aligned in the reverse direction. Sometimes capture data only aligns in one direction. This will affect mutation scores and the condensation tool. Here you can see an example of capture data aligning in one direction. In the NextGENe Viewer, a greater than symbol points towards the 5' end of every read. Normally, the mutation score introduces a penalty for reads that are biased in one direction because the beginning of the reads are more accurate than the ends of the reads. This penalty, the read balance score, can be disabled in the mutation report settings. If condensation is used, the score is disabled by default because the condensation tool can account for most read balance problems. Something else to consider is that some default settings in the condensation tool assume that the data will be mostly directionally balanced. The 3 changes seen here should be made in the advanced settings if the data is one directional. A separate setting requiring some forward and reverse read balance is off by default and should remain disabled for unbalanced data. A second important property is the presence of duplicate reads. These reads occur when PCR is performed after the sequencing library is created by the addition of sequencing adapters. It is recommended that these duplicate reads be combined into single reads in order to avoid errors caused by PCR bias. This can be done using the condensation tool. The advanced settings can be adjusted to keep single copies of duplicate reads and copies of non-duplicate reads, or to only keep the single copies of duplicate reads. If the coverage is high and most reads are duplicates, it may be useful to only retain a single copy of duplicate reads because the remaining reads are unique only because of sequencing errors. If a smaller fraction of reads are duplicates, or if removing duplicates reduces the coverage too much, it makes more sense to retain all non-duplicate reads in addition to keeping a single copy of duplicate reads. Here you can see 2 groups of duplicate reads highlighted in different colors. At least 2 other groups are visible but not highlighted. A read is a duplicate if it has the same sequence and the same number of bases. Duplicate reads are identified separate from the alignment. If and where they match the reference don't matter. Duplicate reads are a problem because amplification bias may alter the allele percentage of mismatches, resulting in false positives and false negatives. In this example, a SNP was present in only 15.47% of reads in the original alignment, and so it was not called. Most of the reads in this region turned out to be duplicate reads, and over 93% of those did not have the SNP. This means the SNP was present at a much higher level, 37.86%, once duplicate reads were combined into single reads. The new allele percentage was high enough to pass the 20% mutation filter, and the SNP was called in that alignment. Here we have an example of the opposite situation, a false positive caused by duplicate reads. At this position, more than 80% of the reads were duplicates. The mismatch barely passed the mutation filter in the original alignment with 24.16%. After removing duplicate reads, it was present less than half as often, 11.11%. Furthermore, the mismatch was associated with another mismatch 9 bases upstream. Every read with a mismatch also had the second mismatch, as long as the read covered both positions. It may be that the mismatches are the result of real data, but were from an inadvertently captured sequence. Specificity is another important property. As many as half of all the reads may have been captured from parts of the genome that were not targeted. It is important to align to the entire genome rather than a reference based only on the regions of interest, because reads captured from outside of the region of interest may be similar enough to map within those regions with a few mismatches. This would create false positives. Specificity can be calculated using the expression report as discussed earlier. Knowing the specificity helps to make a more accurate estimate of coverage across the captured regions in order to optimize settings for the condensation tool. Uniformity is also important. Capture data usually does not perform equally across all regions. Some regions will have coverage much higher than average, while other regions will have little or no coverage. Like specificity, the uniformity can also be assessed using the expression report tool. Regions with low coverage will be missed if the condensation tool is used. The condensation tool is recommended for datasets that have sufficient coverage. It helps solve three of the main problems with next-generation sequencing. It reduces sequencing errors, it lengthens reads so that longer indels can be detected, and it reduces the total data volume to improve processing time, although it does keep the original coverage information. For 454 data, the condensation tool can be used to correct sequencing errors. It is important that there is sufficient coverage. Reads from an area with too little coverage will be saved in a separate file. 30x coverage is close to the minimum coverage necessary, but over 100x is suggested. You can estimate the coverage of your region of interest by multiplying the number of reads by the length to get total base pairs. Divide this by twice the size of your regions of interest. The actual coverage may be higher or lower depending on the specificity, uniformity, and how many reads have aligned. For example, 8,072 base pair reads have about 115x coverage for a 5 megabase capture region. Even if the estimated coverage is very high, it is likely that some of the regions of interest will be missed due to low coverage because there is never perfect uniformity. If the condensation step is selected on the Application Settings panel, then the condensation panel will open while using the project wizard. The Inspect Input Files button will automatically adjust most settings. The expected depth of coverage needs to be manually adjusted because at this point the program has no way of knowing the size of the region of interest. There are three condensation types. Any of the three methods can be used with Illumina and solid data. Only error correction is used with Roche 454 data. Error correction, of course, helps correct sequencing errors. Elongation corrects errors and also lengthens reads. Consolidation corrects errors, lengthens reads, and reduces the overall data volume. Paired information is only retained for elongation and error correction modes. The condensation tool can also be used to combine duplicate reads and single reads. Reads without duplicates can be saved in a separate file or along with the duplicate reads. It may be useful to save the single copy reads separately when most reads are duplicates because their uniqueness is likely due to sequencing error. It is important to note that paired read information is not maintained when duplicate reads are removed. This processing is accomplished using the condensation tool. Condensation must be selected on the Application Settings panel. Consolidation should be selected as a condensation type, and the advanced settings need to be adjusted. First, select the 1 index per read setting in the upper right corner. Some settings will be automatically adjusted. Next, set total reads required for each subgroup to 2. Leaving it set to 0 will keep non-duplicate reads. Finally, select Fixed then Extended Shoulder Length and set the length and score to 8 and 0.01 respectively. Select OK and then click Finish to run the analysis. The single reads representing duplicates are saved in a cycle1.fasta file. Reads that were less than 26 base pair and single reads, if the total read setting was set to 2, are saved in an uncondensed _raw.fasta file. If alignment is performed in the same project, the cycle1.fasta file will be used as a sample. This file should be loaded as a sample in any subsequent projects. Reads that are used in the analysis, usually duplicate reads before removing duplicate reads, are saved as condensed _raw.fasta. In the future, NextGENe will have a standalone tool for removing duplicate reads that will be able to retain paired information.